Hello, students. This is Dr. Faidan Mirza. We are continuing our discussion of coordination and control, and we are trying to understand how nerve impulses are transmitted along the neurons. So, the transmission of nerve impulses along neurons, this is an electrical process. All across the entire length of a neuron, the signal travels as an electrical signal, which we call an impulse. Across the synapse, it's chemically transmitted to the next neuron, and the next neuron becomes capable of producing another nerve impulse. All the neurons, they are capable of generating and transmitting the electrical signal, which is called the impulse. And this electrical signal travels all along the length of the neuron until it reaches the terminal end of the neuron, where the neurotransmitter is then released at the synapse, which then activates the next neuron to carry on the same process. Our nervous system uses various neurotransmitters, and we'll be discussing acetylcholine as an example later on. Electrical signaling is basically generated wherever we have sensory receptors, and these sensory receptors could be at the sensory neuron ending, it could be the dendritic spines of sensory neuron or the relay neuron, it could be the you can say the dendrites at the cell body of the motor neuron as well. So electrical signal is generated at all these places. Here we need to understand a few terms. Potential difference. The term potential difference means the difference of potential between 2 points. So you place one electrode at one point and another electrode at another point, and you try to gauge the difference in the potential of charges between those 2 points. Resting membrane potential, the value of resting membrane potential is about minus 70 millivolts in the human neurons. And this value of minus 70 millivolts is called resting potential because it means that when the neuron is in a resting state and it's not transmitting any electrical signal, any electrical impulse is not being traveled anywhere along that neuron, the neuron will be at a resting potential and the value will be minus 70 millivolts. How is this gauged? How do you record this? When electrical activity is recorded at the neuron, for example, this is a piece of axon with the myelin sheath wrapped. You can see the axon with the cytoplasm inside. The node of Ranviers are there. Myelin sheath can be seen, which are the Schwann cells, mainly the Schwann cells. This is the outside of the cell. This is the inside of that axon. What are we doing? We are just placing electrodes. An electrode is placed at the surface of the axon at the neural lemma, and the other electrode is inserted inside the axon. The electrode placed at the surface is our standard electrode, and the one which is placed inside is the recording electrode. The difference of potential between these 2 points is recorded in the voltmeter. When the neuron is in a resting state, the value seen in this voltmeter in a human neuron would be minus 70 millivolts, which means that your neuron is at a resting potential. For example, this was the reflex arc that we studied. This was the sensory neuron, the relay neuron, the motor neuron, and the effector. These sensory receptors were supposed to respond to the stimulus of pain. For example, when a pin pricks at this point at the finger tip, so you withdraw your hand, your bicep muscle just contracts and you have a withdrawal reflex. Now let's suppose if the pin is not pricked yet, the fingertip at this point, the sensory receptors which are present in the skin, these are the endings of the sensory neuron. All these sensory endings, the sensory receptors, as well as every part of the sensory neuron is in a resting state. It means if you place the electrodes, the surface electrode at the surface, and you insert the recording electrode inside, the value will come out to be minus 70 millivolts. Here, 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 or at any point, even at the region where you are where this neuron ends, even at this point, at the synaptic endings. Since there is no impulse being transmitted, the entire neuron will be at a resting potential. So is the case with the relay neuron. Every part of this relay neuron will be at a resting membrane potential or minus 70 millivolt. Resting potential can also be called as resting membrane potential. So the same thing goes for the motor neuron as well as for the effector. Now, what's an action potential? An action potential is a brief change in the potential difference from the minus 70 millivolt value to a plus 30 millivolt. This is called as depolarization because the cell membrane loses polarity of minus 70 millivolts and shifts to a value which is plus 30 millivolts. And as the value changes from minus 70 millivolts to plus 30 millivolts, an impulse is produced or generated, and this impulse then moves ahead in the neuron. So, for example, if this region, the sensory receptors, are present in the skin at the fingertip and a pin just pricks at this point, this region of the sensory receptor gets stimulated and from minus 70 it shifts to plus 30. The moment it becomes plus 30, it means that this is not as resting membrane potential anymore. This point has become depolarized, whereas the remaining neuron is still at resting potential. The depolarizing wave, or you can say the wave of depolarization of which initiated here because of the shift in the membrane potential from minus 70 value to plus 30 millivolt, caused the impulse to go ahead into the neuron and start traveling along the neuron. So the value of every subsequent part of the neuron keeps becoming plus 30, plus 30, plus 30, plus 30, and the impulse keeps traveling ahead. The previous regions of the neuron from where the impulse have gone forward undergo recovery. And when they undergo recovery, they regain their minus 70 millivolt value again. When the wave of depolarization is traveling and eventually it comes to this point where the sensory neuron is ending, the wave of depolarization arrives at the end of the sensory neuron and the neurotransmitter is released by the sensory neuron ending into the gap which is present between the sensory neuron and the relay neuron. The neurotransmitter released here is in response to the depolarization which is occurring in this region of the neuron. The release of the neurotransmitter here then triggers the relay neuron to become depolarized. So this relay neuron initially was at minus 70 and every part of its entire length was at minus 70 as well. But the moment neurotransmitter got attached at this point, this region became plus 30. It got depolarized and yet remaining neuron was at minus 70. So this became plus 30, then this became plus 30, then this became depolarized, and the wave of depolarization kept traveling all along the relay neuron until it's reached this ending of the relay neuron where it is about to form a synapse with the dendrites of the motor neuron. Again, a neurotransmitter is released here. The release of the neurotransmitter at the synapse is because of the wave of depolarization coming here. The neurotransmitter which is released at the synapse in the gap between the relay neuron and the motor neuron allows the motor neuron dendrites to become depolarized. Now the motor neuron becomes depolarized, its value becomes plus 30. So the value of plus 30, that wave of depolarization travel all along the motor neuron, and this travels all along the axon, eventually reaching the terminal synaptic knobs of the motor neuron. Once the wave of depolarization arrives here, again a neurotransmitter is released that goes on to attach to the effector, which could either be a muscle or a gland. In the reflex arc where we are discussing withdrawal reflex, this must be a muscle. So the neurotransmitter is released at the membrane of the muscle. The muscle membrane also depolarizes in the same way as the neuron was depolarizing. So the muscle membrane was also at a resting membrane potential. The moment the neurotransmitter got released by these endings on the muscle membrane, this muscle undergoes depolarization, and the depolarization of muscle causes the muscle to contract. So this is how action potential begins. So the main things that we have discussed here are that the neuronal signaling is electrical all along its length, and across the synapse it's chemical. There are changes in the potential difference there is resting membrane potential, and there is action potential where the value changes from minus 70 millivolts to plus 30 millivolts. That's it from my side. Thank you so much.